Next up is Ernesto Lima, who's telling us about investigating the effects of hypoxia on immunotherapy response in murine models of colon cancer using mathematical modeling.
Thank you. Good afternoon, everyone. As you already mentioned the title, and the title doesn't roll out of the tongue that easy, not going to repeat it, but I'm going to be investigating immunotherapy. My name is Ernesto Lima. I'm research associate at the University of Texas. And so, I don't know why it's looking that weird, the fonts, but it's good. So this is the outline for the next 12 minutes of this talk. We're gonna go over some of the motivation of our work, why we are studying immunotherapy, and what are the experiments that motivated the developing of the mathematical models that are going to present today. Then I'm going to show the mathematical models itself, the modeling framework, results, very straightforward. Nothing is hidden here. You can see, very easy. Let's go with the immunotherapy. What's the motivation behind this work? Well, we decided to study immunotherapy, and we saw that immunotherapy using checkpoint inhibitors can work well to treat cancer, but not all the patients will respond. A minority of the patients will respond to therapy. And one of the reasons that this happens is because hypoxic tumors generally does not respond very well to immunotherapy. Normoxic tumors will respond better to immunotherapy. One of the solutions is to use a different drug in combination with immunotherapy that can rescue the response in hypoxic tumors. So one of these drugs is ivofosfamide. When the ivofosfamide is delivered together with immunotherapy, hypoxic tumors can respond better to treatment and increase the efficacy of immunotherapy. So the goal of my project is to study the effects of the combination of these two treatments. So what's the experimental framework that we use it? When I say we, please take into account that was one of our collaborators. I didn't do experiments, but I've been heavily using them. So what are the experiments? We have some experiments with mice. We inject the mice with MC38 colorectal cancer cells. We measure the tumor volume over time, and these mice, they receive either immunotherapy or ivofosfamide or a combination of both. We also did F-myso PET, and we track the hypoxic fraction of the tumor. So this was the data. The data is already published and available in this paper here from our collaborators from University of Alabama. And okay, the test is a little messed up. I don't know why. I swear I sent it correctly. But we divided the mice into 6 experimental scenarios. We have one scenario. The first scenario was the control. The next 2 were hypoxic or normoxic tumors that were treated just with immunotherapy. The next one, scenario 4, the mice just received ivofosfamide. And the last 2 scenarios, the mice received immunotherapy and ivofosfamide. So a combination of these 2 treatments, and the tumor could be either normoxic or hypoxic tumor. So we had this data looked great, but now we needed, I'm not going to show the data now. You need to wait to see it a little bit later. But we had this data, and we want to come up with a model to help us understand what's going on with the data. So we develop a model that can track this data. The model, our approach generally is to start with the simplest model that we can, check if the model can capture the behavior of the data. If the model is unable to capture the behavior of the data, we try a different model. So this was the simplest model that we tried. We had a system of ODEs, one equation for the tumor volume, one equation for the well-vascularized fraction of the tumor. So we are measuring hypoxic fraction, 1 minus hypoxic fraction, well-vascularized fraction. So we had these 2 equations for the tumor volume. We are assuming exponential growth. No need for the logistic growth in this case. We have a natural death of the tumor. So we have this mu-T that will capture the natural death of the tumor. And we have an extra term that will capture the response of the tumor to the immunotherapy. So you can see that the effect of this immunotherapy will disappear over time. So there is a certain decay of the effect of the immunotherapy. For the well-vascularized fraction, the maximum fraction is 1. So it's a logistic growth. And we also have a deterioration of the vascularization. So we can assume that as the tumor increases, the hypoxic region of the tumor will increase. So by consequence, the well-vascularized fraction will decrease. And we assume that this other drug, the ivofosfamide, will increase the vascularization of the tumor. So when the vascularization improves, by consequence, it will improve the effect of the immunotherapy. So that was our assumptions. Of course, there are several simplifications as every model, but we decided to try this one and see what we get. With this model, we divided our experimental data in some clusters, and the blue ones, the blue data, scenario 1, 2, and 3, we calibrate the terms in blue. Scenario 4 and 5, we calibrate the effects of ivofosfamide. After we calibrate the whole model, we use the model to predict the last scenario. So basically, the framework that I mentioned so far, develop the model, we saw the model, calibrate scenarios 1, 2, and 3, fix these parameters, calibrate scenario 4 and 5 related with ivofosfamide, and then we predict scenario 6. After prediction, we check, is the error, did we manage to predict scenario 6? Is the error below a threshold? If yes, we stop. So you can imagine, some spoiler, we will not gonna get it. If the error is not good enough, what we do? We find the set of parameters that we need to change in order to capture scenario 6. If we are able to capture scenario 6, then now we stop. If we're not able, then we need to define a new model because our model is unable to capture normoxic tumors with immunotherapy and ivofosfamide. So this is the framework that I'm gonna use, and these are the next steps that I'm gonna show. We're doing a Bayesian calibration. We are assuming all the priors, all the parameters in our model are uniform, have a uniform distribution. So we just assume that we know the range of the parameters. We're using a Gaussian likelihood, and we are using the data from the tumor volume that we measure, I think 14 times. And we are using the data from the hypoxic fraction that we are converting to well-vascularized fraction. So as I mentioned, first we calibrated the first 3 scenarios. The top row is the tumor volume. The bottom row is the well-vascularized fraction. And we have scenario 1, 2, and 3. And you can see that our model was pretty good to fit all of the scenarios. So we are able, one thing that I should point out, that differently from what we usually do, we are not doing a mouse-specific calibration, but we are doing a whole population calibration. So all of these 3 parameters, we were able to reproduce these 3 scenarios with the same values for the parameters. So we fitted that, but we don't have the calibration for the parameters related with ivofosfamide. So with these parameters, we calibrated scenario 4 and 5. Also, we were able to, using the parameters that we had before, to find values that we would be able to capture these 2 scenarios. The next step, let's predict scenario 6. See if, knowing the values that we have for the other parameters, if we can predict this scenario. Of course, we were unable. So what should we do? We developed the model, we could not capture. Let's see if we can try to find one set of parameters that can capture. Let's try to fix the most number of parameters that we can, and let's find the parameter that we need to change. To find the set of parameters that we need to change, what we did a sensitivity analysis to rank the most important parameters for our model. What I mean by most important if you are unaware of sensitivity analysis, basically small perturbation on these parameters will have the huge impact on our quantity of interest, on the tumor volume. If you have no clue about sensitivity analysis, is what you need to know from this figure is basically the highest the value here of the total effect index, the most important is the parameter. So we can see the most important parameter is gamma I, which is the death rate due to immunotherapy, followed by the proliferation rate and the death rate of the tumor. So we decided, let's see if fixing all the other parameters and changing just this value here of gamma I, can we reproduce scenario 6? Yes, we could. So we changed just one parameter and we were able to go from this to this by just changing one parameter. We had to increase a lot. And what we concluded from that? We concluded that in our assumption from the model, from what we had on what we observed on the literature, we had used before that ivoflosamide was just increasing the vascularization, and we were hoping that this would be enough to capture the 6 scenarios. But what we noticed from the modeling and from the results that we got, that we also needed to take into account that ivoflosamide not only increased vascularization, but also sensitize the tumor to the immunotherapy. So increase the efficacy of the immunotherapy and increase the response of the tumor to the immunotherapy. So in summary, we develop a system of ODEs that can capture tumor and vascularization response to the combination of these 2 drugs. We also note that we are able to capture the scenario 6, which is normoxic tumor with the combination of these 2 treatments, when we assume the direct effect of ivoflosamide on the death rate by the immunotherapy. So the next step, we are doing other experiments with different combination protocols because our goal is to optimize the delivery of these 2 treatments to find the optimal response for that. So I would like to thank you all my collaborators. Here are the fundings, here are the collaborators. They are the ones that did all the amazing experiments that we are using. They're from the University of Alabama, and here's Tony and Claude from the University of Texas. Thanks so much.
How did you figure out what to adjust that parameter by?
We did the calibration. We just did the calibration, fixing the other parameters and let that one free to calibrate it. We actually, we got, not got lucky. The first parameter that is the most important parameter was the one that solved it, but we, just for the sake of sanity, we tested all the other parameters to see if changing the other ones, we would be able to recover the other scenario, but we were unable. So we try a leave one out approach where we fixed 5 parameters and recalibrated one of them. And this was the only one that was able to, that we need to change to capture that scenario.
Thanks.
Malcolm.
Thank you. I was also going to ask about something similar, but in addition then, I was wondering, what's the justification then for altering the parameters before looking at altering any of the model terms?
We could have, our goal was to keep the model as simple as possible. We could have, for example, the death rate by the death rate with the immunotherapy in function of the concentration of ivoflosamide or in function of a more complex term, but we decided to try first to see if the model itself would be able to capture the scenarios by just changing the value instead of coming up with a more sophisticated function for this effect.
Okay, so because I was just wondering if maybe changing the model could predict the same sort of results at the beginning, but then with a less drastic parameter change, something along those lines.
Could be. The change on that parameter specific was, we had to increase by 18 times, but the total of that rate we increased by 2.5. So, which is not that bad.
Thank you so much.
Okay.